everyone, welcome to the latest A Spotlight On interview. Today, I'm joined by Kimberly Gilmour, and we're going to be talking about variants of uncertain significance. Kimberly, if you could please introduce yourself and tell everyone a little bit about what you do.
I'm Kimberly Gilmour. I'm Director of Laboratory Medicine at Great Ormond Street Hospital. My personal focus and focus of our laboratory is investigating patients with genetic defects of their immune system. And one of the things we found quite recently is that a lot of these patients develop what we call or we identify variants of unknown significance. And those variants of unknown significance say exactly what it says on the tin. They're a change in someone's DNA, and we don't understand if it's pathological and causes disease or if it's benign and it doesn't cause disease. And my whole career is sort of led up to trying to set up tests to functionally validate these and identify, are they benign or are they pathogenic? So I did a PhD at the State University of Stony Brook in Long Island. After I finished my PhD, I moved over to what was then the Imperial Cancer Research Fund, now known as Cancer Research UK, where I did a research fellowship. And from there, I was hired by Great Ormond Street to actually take my PhD work and turn it into clinical diagnostics. And for the last 22 years, I've been doing that. And I was hired to set up what was initially diagnosing children with primary immune deficiencies. And the way we used to do it before next generation sequencing was we would screen for protein expression or function. If it was missing, we would know to sequence that single gene, because we didn't have the ability pre the whole genome sequence to sequence the whole genome, to look at lots of genes. So we tried to do very targeted genetics. And then as things moved along, we had the advent of next generation sequencing and whole exome and whole genome sequencing. We then almost flipped the paradigm. We would find lots of mutations or variants, not know if they were pathological or not, and then develop the functional tests to see if they were disease causing or not.
Fascinating. And you've given a definition there. Are the definitions of variants of uncertain significance, are they consistent? Do they vary? And what's the prevalence of these variants?
So both the United States and in Europe, there is a classification of DNA changes. So they go from benign, and we know they're benign because 20% of the population has them, and obviously not 20% of the population has these very rare diseases, to likely benign, present in the population at a lower level, but still common enough to be unlikely to be disease causing. Variants of unknown significance, meaning we don't know if it causes disease or not. We really don't know, to likely pathogenic. They're rare variants, they're associated with disease, they prevent protein function, they're likely to be causing disease, to certainly disease causing. Ones that have been proven in multiple families from multiple ethnicities or different backgrounds, and they cause disease. So it's that range of changes. Unfortunately, a lot of changes end up in that middle batch, and it's very specific on different genes, because some genes we have a lot of information on. They've been sequenced many, many times. We've seen them in lots of populations, and they're very well classified. So you don't get very many variants in those particular disease genes. Cystic fibrosis, we get fewer variants because it's a disease we've known for a long time. We've been sequencing the CF gene since the 1990s. We initially knew about hotspots. Now we sequence the whole thing. We have some good functional tests to go with it. The more you know about a gene, the fewer variants of unknown significance you get, because every time you get a change, ideally you classify it as disease causing or benign, and you can only have so many changes. The genes are only so big. Rarer diseases, you get many more variants because we know much less about the disease. So it is somewhat genotypic, gene type specific in terms of variants. If you just look at a human being and you sequence their whole genome, you can get as many as 30,000 changes. But most of those are going to be benign, and they're not going to cause disease. And it's filtering down to the ones that are likely or potentially pathogenic. So a lot of times we only look for variants in the genes of interest. We don't look for variants in genes like that cause hair color or eye color, because we don't actually care about those.
That makes sense. And you talked a bit about our current ways of functionally validating these variants from protein expression and protein function. What are some of the challenges of our current methods?
Okay, so initially almost all variants run through what's called in silico or computer-based analysis. And that gives you predictions if they're likely to be pathogenic or not. And we think in general that a stop mutation where the protein's not made to its full length is likely to be pathogenic, for example. A big deletion, if you chop out a bunch of the protein, is likely to be pathogenic. If it's an amino acid change, but it's conserved from zebrafish to humans, and everything from zebrafish to humans has that same amino acid at that position, it's likely to be pathogenic, or that's a big hint it might be pathogenic. If it's very rare in the population, it's likely to be pathogenic, because it's really common. We're not all wandering around with these rare diseases. So all of those are big hints that they're pathogenic, but it doesn't tell you for sure. So you really have to do something else. And you can look at RNA, because if you don't make RNA, you're not going to make protein, and therefore it's likely pathogenic. That's very useful for splicing defects or defects that might affect transcription of a protein, of the message and then the protein. If it causes, you can then look for protein expression by immunoblot, by flow cytometry, by microscopy. If the protein's not there, then you have a very big hint it's pathogenic. But a lot of proteins are made but non-functional. You've changed a single amino acid and it's essential for a signal transduction pathway or sending messages between cells, but the rest of the protein's there, it just can't work correctly. And in that case, it's very difficult because you have to do a functional assay. And to do a functional assay, the biggest challenge is having the material to do it. So you only can look at where that protein is expressed or where that message is made. So I'm an immunologist. It's relatively easy for me. I'm interested in white blood cells. Assuming someone hasn't had a bone marrow transplant, we usually can get a sample and do that because that's very easy. But if you're a neuroscientist and you're interested in brain signaling, that's very hard because you're not going to be able to just take a little piece of someone's brain to see if it's a functional mutation or not, in which case you're much more dependent on animal models or cell culture models, tissue culture models to try and do it. More and more, we're using induced pluripotent stem cells. So you can take a biopsy, make fibroblasts, and then turn them back into stem cells and then redifferentiate them into what you want and try and do function there. But that's really complex and not very efficient because we're now doing whole genome sequencing at scale within the NHS in the UK. So we have lots of patients going through this. And those are really interesting for new genes or one-off assays, but they're not useful when you have lots of patients coming through. So if we can take tests we're already running in clinical labs or develop tests that can be run at scale in clinical labs, that's much more useful for the patients.
Fab. And what are the benefits of whole genome sequencing here, and what are the limitations as well?
Okay. The benefit and the limitation are almost the same thing. So the benefit is you get so much data. So if you do whole genome sequencing, not only are you looking at the exons, the coding pieces of the gene, but you're looking at all the other areas that help control gene expression. So you're looking at promoters and enhancers and splicing machinery and all of those things. So you have a much greater rate of picking up disease-causing mutations, which is great for patients who may have been undiagnosed for many years. But you also pick up many more variants. So there's a lot more, and they're even less likely for the in silico, for the computer program to be able to model, because we're much better at modeling amino acid changes than we are something sitting in a promoter site. And the functional validation becomes much more important because you don't get those clues necessarily from the in silico. So the benefit, we find many more mutations, we help diagnose patients who haven't been diagnosed before, is also the drawback because we get all these extra variants that you have to functionally validate.
And what data do we need in order to identify variants more effectively then? So you talk about patient samples. Are we also looking at family samples, general population samples?
So it's really, so a lot of our algorithms in terms of identifying the variants are based on the fact that you have a child who might be very unwell and parents have had the child, so you assume that they're fairly well. So you're looking for things that the parents are heterozygous for and the patients are homozygous for, so they've inherited a bad copy of the gene from each parent, and that's really helpful. Or things that are de novo, neither parent has it and the big child has it. You still miss a lot of mutations that way. You miss things that might be partially penetrant, so the parent may have it but doesn't get really sick with that change, and the child does get really sick with it. So that having trios or family groups is really helpful, but in and of itself doesn't necessarily solve the problem. I think the bigger thing that we're trying to work with our colleagues across Europe and within the United States is we need much better data storage and data banks, so that if a lab in London says, this is definitely pathological, here's our variants, then a lab in France doesn't have to redo that assay, or a lab in the United States. Someone's already shown it, and you have that evidence. or you know exactly what assay was done, so you could just repeat that one assay rather than trying different things. There's really good ways of recording your variants, but there's not really good ways of recording the functional data that may have been done, and it's used to reclassify those variants. And I think particularly we're really good about publishing and sharing positive findings. Definitely disease causing, no proteins made. You do a little case study, you write it up, you do something with it. Completely normal protein, completely normal function. If it's completely normal, people tend not to share that information as much, but that's equally important because you don't want to waste someone's time trying to prove it's pathological when actually it's completely benign and the cells do everything they're supposed to do. And we're less good at sharing that because that doesn't get published. That's not as exciting. You're less likely to get grant money for that.
So publish or perish strikes again.
Still is, exactly.
Earlier this year, we saw a biotech company announce their $100 genome. What technological advances have either already happened or you think around the corner that could advance your work?
I think we're really good at sequencing, and sequencing has become cheaper and cheaper. So you can get a sequence for $100. It's the analysis and interpretation of that data. And I think as artificial intelligence gets better at it, and we get better at sharing that data, we'll have a bigger data pool or data banks to draw from. I think that's really where it's going to be the artificial intelligence to analyze those genomes so that you pull out what's likely to be pathogenic. But it does depend on the algorithms used. So we had a case recently where we found one disease in a patient, but actually it looked like something else was going on as well. But the algorithms used assumed it was autosomal recessive. So you had to have a bad copy from mom, a bad copy from dad. So baby gets 2 bad copies. That's why they were sick. That identified the first disease in the baby. But we missed the second disease till we went back and did some better family history. It turns out we'd excluded some genes because mom was homozygous for it. But when we went back, mom also was a disease that was involved in height and things. Mom was very tiny, and actually mom was affected. But the algorithms being used by the computers had excluded that gene because mom was homozygous, so she couldn't have it, even though she did have it. So I think the problem with artificial intelligence is it's how those algorithms are written, and to understand how they're written. So if you start looking beyond that, you know what you're looking for.
Absolutely. And empowering the patient often also requires more involvement from them as well. Obviously, this is quite intense family focused, patient-led medicine. So what challenges do you see as we shift our systems to patient-led with these complex diagnoses? What can we learn from the way that you are diagnosing and treating?
I think it's really important to have patient and parent involvement because they are the ones giving the samples. It's their material, it's their DNA. They're going to benefit potentially from those diagnoses, but those diagnoses also can be life-changing, and some people don't necessarily want to know that information. So I think it's really important to be really clear with families and patients what you are and are not looking for. So are you just going to do whole genome sequencing, but you're only going to look at a panel of genes? Are you going to look for, are you report back variants or changes in genes that aren't related to the disease you're looking for? Are you going to report back what are called actionable findings? What if you sequence for immunodeficiency but find a BRCA1 mutation? Are you going to report that back? I think there's a lot of ethics around that. And I think families really have to understand what you are and aren't looking for, what the limitations of the data are. I think families, we've had several that have consented to reanalysis of that data, and things that we ran 2012, 2015, we didn't know as much. So we didn't look for the things we're looking for now, and reanalyzing that data has led to diagnosis in some of these patients. As we move to gene therapy, this is actually really important, because if you're doing gene therapy, it's essential that you know that mutation is disease causing, so you put back the right gene. And at Great Ormond Street, we've treated a lot of our patients with gene therapy already.
And you've just talked about Great Ormond Street. Obviously, that's been a huge chunk of your professional and personal history as well. Can you tell us about the history of the hospital and their cell and gene therapy history?
Okay. So Great Ormond Street is the oldest children's only hospital in the United Kingdom. It was founded, I'll get it probably wrong, but around 1858. So very long time ago, has always been a charitable hospital, has always been only children treated. But it was a hospital that very early on started doing bone marrow transplants, not only for hematology in the pediatric leukemias and things like that, but for these rare immunodeficiencies, and has had a real interest in immunodeficiency along with our collaborators at UCL Institute of Child Health. And together, a gene therapy program was developed, led primarily by Professor Adrian Thrasher, who's still at ICH. And our first patient was treated with XKID gene therapy at Great Ormond Street 22 years ago. And he's now done really well and is an adult. And from that very rare disease, X-linked common gamma chain SCID, so severe combined immunodeficiency, the boys in the bubble, so children who have to be isolated until they either have a bone marrow transplant or gene therapy. That program's expanded significantly, and we treat a number of other primary immunodeficiencies, and we're beginning to treat some metabolic diseases as well. So that's all very much personalized medicine. You identify the defective gene, and if we can do it, we replace that defective gene. And it's all somatic gene therapy. We're only replacing genes in blood cells or in tissue cells, not sperm and egg. And the kids that have had it have done really well. And that's beginning to expand into hematology. So you're beginning to treat things like beta thalassemia and other diseases as well.
And can you run us through one of your, no, is favorite the right word? Can you run us through a really impactful case study?
Yeah, so if we go back, one of our patients was on intensive care. We were able to fairly rapidly diagnose them with XKID in terms of they had no gamma chain protein. The signaling pathways below that weren't working, but we couldn't find the mutation. But they didn't have a good donor. So we didn't have a good donor for a bone marrow transplant. For gene therapy at the time, you still are, you're required to identify the defective gene to do gene therapy. And we couldn't identify the defective gene, although we knew they didn't make RNA for common gamma chain. They didn't have protein for common gamma chain. All the signaling was not working. We went back and did some very old genetics. We did what was called X inactivation. So mothers of this particular disease, their T cells are non-randomly X inactivated. They only develop T cells from the good X. And we could show that mom had non-random X inactivation of the T cells. We could show the child inherited the inactive X. So we had a lot of data. This was common gamma chain SCID, but we couldn't find the mutation. In fact, the parents were very educated and chose to have gene therapy. They were counseled that there was a risk that we were putting in the wrong gene, but there was limited other options. So we did gene therapy. The child, in fact, has done very well. They must be about 18 now, which is brilliant. So they've done really well. But it was about 6 or 8 weeks after they started getting a new immune system. So we knew we'd put the right gene in because we did an in vivo complementation assay, those old-fashioned assays where you put the gene back and does it work again. And then we found the mutation. And then because it was actually the reverse of what we're doing now, we had all the functional data. So we were convinced it was the right mutation. It was actually down in the polyadenylation site, which controls RNA stability. So although the coding region was okay, it was a mutation that controlled the stability of the RNA, and therefore you had unstable RNA, you didn't make protein, and the cells couldn't develop and function properly.
That's a fascinating case study. Thank you so much for sharing. And if we look to the future, what do you think is needed to streamline clinical implementation equitably, whether that's in the UK or internationally? We've talked about data sharing briefly.
I think data sharing is important. I think patient education is really important because patient and parents don't always come forward, or they're concerned about who's going to control their data. I think there's some misunderstanding in some communities. So I think there's a huge piece of work about education across patients around the world, so they understand what genetic sequencing can and can't do. I think we need real clarity as to who owns the data. I don't think you can patent a human genome. I think people should own their own genetic data. And then they can share it with their healthcare professionals. But that's not necessarily how it works in all the different countries. I think that needs real clarification. I think outside of places like the UK, a lot of genetic sequencing is on a paid-for basis. So you have to be wealthy to do it, even though I appreciate 100 genomes out there, quality and analysis and other things are tied in with that. And obviously 100 pounds or dollars is still a huge amount of money if you're in a less developed country. So I think the technology is going to continue to get cheaper and cheaper every few years. The analysis, we need to get more efficient to get it really available to people outside more developed countries. And even within more developed countries, there's an equity issue about who's willing to have it and who's not, or who's scared of having it. Someone's going to know about me, someone's going to do something with my data. So there's a big educational piece, a discussion to be had with the whole community, not just patients and parents, but government, regulators. How do we prevent it being hacked? Your DNA is your ID. Is that ever going to be used as such? There's a lot of questions, I think, that need ethical discussion, much more so maybe than the scientific discussion.
And what are your thoughts on the democratization of genomic data?
I think it's really important, and I think people should be able to access their data. They should be able to understand their data, and that data, if they're happy in an anonymous way, should be shared so other people can benefit, even if it, as we said, if it's things like, this is definitely a pathological variant, this is definitely a benign variant. I think all of that's hugely important. I think it does come back down to education. And I think people still think, oh, I've had my whole genome sequenced, nothing was found. It's not a genetic disease. People don't understand the limits of the technology still and the analysis. So we're really good at sequencing. We can do the whole genome. That's not a problem. We can say it's AATTCCGATC. We can do that. That's not the problem. The problem is understanding what it means, and particularly what all the non-coding space means. And remembering the vast majority, 97+% of the human genome is non-coding. It's all regulatory. We don't even understand what a lot of it does. So I think having people understand the bits that we don't understand is really important too.
Absolutely. And you mentioned biosecurity there as well. What do you think is a key thing that people should take away as we set up these biobanks, both national ones as well as international consortiums? What's the number one thing we need to be considered, whether that's ethically or from a biosecurity perspective?
I think we need to make sure that that data is truly anonymized when it's put in there. Depending on how it's done, you may want it reversibly anonymized, because you may want to be able to say that, oh my gosh, this person has this disease and we can treat it. So if we find out, we can make a difference and save their life. There are genes like that. But you don't want any researchers or pharmaceutical companies or anyone else having anything other than that generic data, with very minimal, you can say they're a, 50-year-old male, and that's about what you would say. The problem is you look at that gene and you might be able to immediately say, well, they're from this ethnic group, and they're predisposed to heart disease and diabetes. And in the UK, it's less of an issue because we don't worry so much about insurance, and our insurance is not dependent on your genetic data. But in countries where they want genetic data to get health insurance or life insurance, I think that's really, you can't do that, because otherwise no one's going to, if you can't get health insurance, if they find something bad on your genetic sequence, would you ever have a genetic sequence done? If you can't get life insurance, and you need life insurance because you have small children or whatever, and you can't get it if you have, a predisposition of diabetes or whatever, well, would you actually ever be sequenced? But if you knew you had a predisposition to diabetes, maybe you would lose weight, be more active, change your diet, and never develop the diabetes. So you get into this very complex system, which I don't think is helpful. So I think there needs to be real clarity around how that data is used by commercial providers of insurance and things like that.
Absolutely. And very optimistically, what are your hopes for the future as we learn more about these variants of uncertain significance?
I think we have fewer and fewer variants of unknown significance because we've classified them as benign or pathogenic. That would be huge. So parents and families don't get this, well, we found this and we don't know what it means, or it's never reported to them, but actually maybe it's pathological and we haven't reported it, but we probably should have because we didn't know it was pathological. So I think, in 5 to 10 years, there may be no variants of unknown significance. We might have, at least in the coding space. In the non-coding space, I think it will still be a lot of information to learn, but in the coding space, that would be huge. I think for families, you have a sick child, you come into intensive care, we can get a sequence back in 2 weeks that's diagnostic and you get optimal care. That we're beginning to do that. But the more we do that, I think that would be amazing. I do think people have to understand that genetics are not the whole thing. There's still a huge environmental component. So COVID being the classic example, it doesn't really matter what your genes are. People of every genetic, very few people have not gotten COVID at this point. We have genes that may say you may get sicker with COVID. We haven't really found any good genes that say you don't get COVID. And if COVID didn't exist, we wouldn't have had all these people in hospital and things like that. So your genotype didn't really matter particularly. I mean, for maybe a very, very small percentage, you were more susceptible to it. You got sicker with it. But for most people, it was because you had a virus. And, there's also some fatality of, oh, I have a genetic predisposition for heart disease, I'm going to get a heart attack. Well, actually, you have a genetic predisposition for heart disease. Why don't you lose some weight, exercise, take blood pressure medicine? There are other things you can do to prevent that from happening. There are genes that say, you have a gene for SCID, you have no T cells, you need a bone marrow transplant or gene therapy. That is an absolute. But a lot of genetics isn't that absolute. It's a combination of genes and environmental factors. So it's still remembering all those environmental factors and all the things we can do ourselves to take care of our own health.
Absolutely. And what is the most common answer you actually get when you're validating a variant of uncertain significance? Are you getting more, yes, this is pathological, no, okay, it's benign, or a lot of maybes?
We get a lot more definites than maybes. Part of it is, I'll be really honest, we're screening a lot in silico. So we're taking the ones that have a higher probability, they're rare, they're more conserved, it's never been seen before, and it fits with the phenotype. So we're looking at variants in a set of genes that we have a particular interest in because they cause immune diseases, and it fits the phenotype. And most of those, maybe 70%, we are confirming is pathological, but it's a selected group of variants. We're not looking at every variant the child has. We're looking at the variants in the genes that are likely to contribute to their phenotype. There are certain genes that just have a lot of variants that are benign. So there are a couple of genes, and you have to be careful that you just don't go, oh, it's just another variant in that gene, because you do actually have to look enough to think, well, would that fit? Could that be? How common, rare is it? Is it worth following up? So I think at the moment we're not self-selecting, we're choosing which ones to follow up because we don't have the resources to follow up all of them.
That makes sense. And are you left feeling at the end of the day like Sherlock Holmes or Miss Marple having solved the case?
When you solve the case, it's really exciting, particularly if you solve it in a way that's really unexpected. So a couple of years ago, a colleague of mine who's a mitochondrial, Shamima Rahman's a mitochondrial physician, came to me and said, well, we have this family and we thought they had a mitochondrial disease. And we looked at their mitochondrial genes and didn't find anything. So we did whole exome sequencing. And the only thing we found was this variant in a gene called STAT2. And STAT2 is an immune gene. It's involved in immune signaling. And she's going, I don't understand, but it's the only gene we found. And both siblings are homozygous for it. The parents are heterozygous. Genetically it fit. But why is it causing mitochondrial disease? And in particular, these patients had really long mitochondria. So in a normal human being, your mitochondria they grow, then they fission, they split in half, and then they grow again and they split in half. So they're either small or they get to a little bit bigger, but then they split in half and they keep going through this cycle. And these kids just had really long mitochondria. They were never fissioning. They were never going through the cycle. They were just getting bigger, which was really, and so they definitely had a mitochondrial disease. You could see this. You could see it by electron microscopy. You could see it by confocal microscopy. But the only gene we found was an immune gene. And then we actually did a lot of work. We did complementation assays. And if we put the correct gene back into those cells, their mitochondria then went through the cycle of growing and fissioning and growing and fissioning. And in fact, we were then able to go a bit further and show that there was a phosphorylation defect in a particular protein in the mitochondria that this immune gene, STAT2, was controlling. So that was really exciting. And you did feel like absolutely like Sherlock Holmes, because you'd taken this case that didn't make any sense at all. And so it was a mitochondrial disease with an immune gene and showed how they work together. And I think we're going to see a lot more of that. I think there's a lot more interaction of genes that we all get in our little silos, but actually we need to work much more together. So that was a really exciting experience.
Thank you so much, Kimberly. I've learned so much and completely agree that the importance of null results is just as valid as everything else that we've talked about today. And I'm very jealous that you get to play Sherlock Holmes so often. And thank you for joining us today.
Thank you very much, Poppy.
Hello everyone. If you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website, frontlinegenomics.com. I hope you enjoy.